Have you ever stopped to think about why two people can eat the same foods, take the same supplements, and still have very different outcomes inside their bodies? Maybe you've noticed that what seems to work for one person doesn't quite work the same way for another. Today, in this video, I'm going to explain how researchers are studying exactly that question through a clinical trial that looks at how the body processes vitamin E in normal weight and obese women, and why those differences might matter more than we realize. Vitamin E is one of those nutrients many people recognize by name, yet very few truly understand how it behaves once it enters the body. It's found in foods like nuts, seeds, and vegetable oils, and it's also one of the most commonly used dietary supplements. Despite that familiarity, there are still unanswered questions about how vitamin E is absorbed, distributed, stored, and broken down, especially in women with different body compositions. And here's the part most people don't talk about. Much of the foundational research on vitamin E metabolism was historically conducted in men, leaving important gaps when it comes to women's physiology. This matters because women experience unique metabolic and hormonal patterns, and body fat plays a significant role in how certain nutrients behave. Vitamin E is fat-soluble, meaning it dissolves in fat rather than water. That raises a fundamental question. If a nutrient is stored in fat, does having more body fat change how much of that nutrient is available to the rest of the body? Researchers are studying this question because women with overweight or obesity often have higher levels of oxidative stress and inflammation, and vitamin E's antioxidant properties make it a nutrient of particular interest in this context. Yet current dietary guidelines generally assume that absorption and utilization are relatively consistent across body types, which may oversimplify a very complex biological process. This is where the clinical trial titled Vitamin E Pharmacokinetics and Biomarkers in Normal and Obese Women comes in. Researchers are studying alpha-tocopherol, the primary form of vitamin E found in human tissues, to better understand how it moves through the body. Pharmacokinetics is simply the science of tracking what happens to a substance after it enters the body, including how it is absorbed, where it goes, how long it stays, and how it is eventually eliminated. Early research suggests that body composition, meal composition, and even the presence of other nutrients may influence these processes, but those relationships haven't been fully mapped out in women. To explore this, scientists designed a Phase 1 clinical trial. Phase 1 studies are not about proving that something treats or cures a disease. Instead, they focus on understanding biological behavior and safety. In plain terms, this kind of trial asks foundational questions like how a nutrient behaves in the body under carefully controlled conditions. The goal is precision, not promises. But here's something many people don't realize. Without studies like this, later-stage research simply wouldn't have a reliable foundation to build on. Women between the ages of 18 and 55 are eligible to participate, representing a range of body mass index categories, including normal weight, overweight, and obese women. Some participants also include overweight women with mild, non-insulin-dependent type 2 diabetes, provided they meet specific health criteria. These criteria help ensure that the results reflect differences in body composition rather than unrelated medical conditions. This careful selection allows researchers to focus on metabolic differences without unnecessary confounding factors. During the study, participants receive vitamin E through both oral and intravenous administration. Oral dosing reflects how people typically consume vitamin E through food or supplements, while intravenous dosing allows researchers to bypass digestion and directly measure how vitamin E behaves once it enters the bloodstream. By comparing these two methods, scientists can more accurately determine how much vitamin E is absorbed from the gut and how quickly it appears and disappears in circulation. This is one of the most informative aspects of the study design, but it's also one of the least talked about outside scientific circles. Meals provided during the trial vary in fat content because dietary fat is known to influence the absorption of fat-soluble vitamins. Researchers are measuring how different fat levels affect vitamin E uptake and whether this effect differs between women with different body compositions. Blood samples are collected over time to measure vitamin E levels, along with related biomarkers that help contextualize metabolic activity. In some parts of the study, adipose tissue samples are also analyzed to understand how much vitamin E is stored in fat tissue itself. This helps researchers build a clearer picture of turnover kinetics, which simply refers to how quickly vitamin E moves in and out of different compartments in the body. Another intriguing element of this research involves vitamin C. Vitamin C, or ascorbic acid, is a water-soluble antioxidant that may interact with vitamin E metabolism. Researchers are studying whether different vitamin C levels influence how alpha-tocopherol behaves in the body. Participants may undergo periods where vitamin C levels are intentionally adjusted before vitamin E administration. This allows scientists to observe potential interactions without making assumptions. But here's the part most people miss. Studying these interactions does not mean recommending changes to anyone's diet. It's about observation, not instruction. So what do the results show so far? As with many early phase trials, the findings focus on patterns rather than conclusions. Researchers are analyzing differences in absorption rates, circulating levels, and tissue distribution between normal weight and obese participants. Early data suggests there may be variations in how vitamin E is handled depending on body composition and dietary context. but these observations are part of an ongoing process. Phase 1 trials are designed to raise better questions, not to deliver final answers. Looking ahead, there are several possible outcomes. In a best-case scenario, this research could clarify whether vitamin E requirements differ meaningfully across body types, helping inform future nutritional research and guideline development. Even if the differences turn out to be smaller than expected, that information still matters. Neutral results help refine scientific understanding and prevent unnecessary assumptions from shaping future studies. And if some hypotheses are not supported, that's equally valuable. In science, learning what does not appear to make a difference is just as important as discovering what does. Throughout the trial, safety and ethics remain central. The study is overseen by institutional review boards that evaluate risks and ensure participant protection. Informed consent is obtained before participation begins, and monitoring continues throughout the study. Participants are free to withdraw at any time, which is a fundamental principle of ethical clinical research. This deliberate pace may seem slow, but it's one of the reasons clinical trial data is trusted and reproducible. In the end, this study represents a careful attempt to deepen our understanding of how a common nutrient behaves in women with different physiological profiles. Researchers are studying vitamin E, pharmacokinetics, and biomarkers to build a more complete picture of nutrient metabolism, one that acknowledges biological diversity rather than assuming uniformity. This video is for educational purposes only and is not intended as medical advice. As research continues, studies like this help lay the groundwork for more personalized approaches to nutrition. If you're interested in how clinical trials expand what we know about health in the human body, consider subscribing to stay updated on future research explained clearly and responsibly.